everyone. So my name is Cassereau, PhD in Bioengineering, and I actually did my PhD here. And I am now a scientist with Xcell Biosciences. We're based out of Bears Collaborator Space across the street. Thank you for the organizers for giving me a chance to share a bit of my own background, as well as what we're doing as a company, which we certainly think has been quite an innovative process so far. To give a bit of context of where I come from, so I actually did my PhD in this program here. I actually only was about 6 months ago that I defended. And I did my work with Dr. Valerie Weber. And then so that was a mix of sort of cell biology and tissue engineering, in essence trying to understand how a tumor interacted with its environment by recreating those systems ex vivo, essentially allowing you to tune the different things a tumor cell might be exposed to and sort of external to it, and how that influenced its behavior and biology. And that expertise directly led me to pursue a position with Xcell Biosciences. So I joined Xcell Biosciences literally 2 days after I defended my thesis. This was a weekend. And Xcell is a company that was really focused on enabling primary cell culture, very much what I was trying to do to just understand how a tumor interacted with its environment. Xcell's goal was to use that same biology to enable primary culture, so recreating the microenvironment in a way that would allow you to more readily do primary cell culture. And so my role as a scientist and sort of a bioengineering background is similar to what I did for my dissertation work, but a bit of a mix now in this startup environment in which I'm both doing fundamental research and development around different application spaces for our company, as well as then commercializing and productizing those in a way that's meaningful as a tool towards others. And so really getting to the idea of primary cell culture, what the company we're trying to do is reproduce the miracle that was Henrietta Lacks's cells. So Henrietta Lacks, or the original patient from which HeLa cells were derived, was the first human cell line that was actually able to grow ex vivo. If you look at back sort of 65 years ago, the major challenge of cell biology was not necessarily understanding human cell biology, but even being able to do human cell culture. In essence, lucking into being able to get her cells to grow relative to hundreds of other attempts. This alone, those one patient cells were used to generate both the polio vaccine as well as eventually the discovery of human papillomavirus as the driver of her cancer, eventually leading to a vaccine and ultimately a Nobel Prize. So from a single patient cells, all of that value is captured both to the field of cell biology as well as public health. And so as a company, we're really saying, well, there's incredible value in being able to do this more reliably, but in that, why is that it can't be done more reliably today, knowing that 65 years ago it was such a monumentous event? And part of the problem is the techniques which people were trying to use and just hoping to get lucky with 60, 50 years ago haven't changed. We're still doing culture on rigid petri dish style vessels, animal containing serum, as well as in sort of standard CO2 incubators, consistent 5% CO2, humidified environment, regulation of temperature. However, at Xcell, we think of this type of culture similar to trying to grow a fish out of water. Yes, you can see it's a fish, you can tell its shape, you can tell its components, but it's dying. It's not acting as it would in the body, and it's certainly not robustly growing or acting as it would. And when you can, in fact, get it to take to culture, ultimately you're selecting some subset that can survive in this radically different environment, thus sort of driving an evolution that you're no longer really studying the source material you're interested in to begin with. So the question for us began like, well, what if we just created a better tank? Right? What if we created an aquarium in which we can actually grow this fish, or in this case the cell, in an environment that's similar to what it would be exposed to, which should allow us then to actually better understand its behavior in a physiologically relevant way, so better, more accurately targeted drugs, fewer clinical trial failures, being able to actually grow the patient's cells for therapeutic value in and of itself, as well as monitor the patient's response to treatment, and that we're able to sample their own cells. The question became, all right, well, what is it that we're lacking in the current approaches that we would need to better faithfully recreate the physiological environment the cell is exposed to? And an example of one that's just so obvious is oxygen itself, right? Growing a cell in the atmosphere is nowhere near what a cell would ever be exposed to physiologically. However, what also becomes apparent is it's not enough to just introduce the element of control of oxygen into cell culture so much as it is to do it in a tunable way. Cells are not only exposed to much lower oxygens than we have in the atmosphere, but also a range across different tissue types. Cells in the bone have certainly lower than maybe other healthy tissues such as the lung, disease state introduces even more complications, where you might have quite low, say, the core of a tumor. So to capture that, we created sort of a family of products in which we have both a human recombinant-based media solution, which has sort of tissue-specific type supplements, as well as a sort of tunable substrate that allows you to present the tissue structure, organization, other components that a cell will grow on, as well as our sort of next generation incubator 2.0, in which we can control both the atmospheric conditions, or the gas mixture, as well as the pressure that a cell would be exposed to. And importantly, we've had success with this sort of composite of products in a variety of different applications. I want to share a few. Specifically looking at growing a patient's own tumor. So on the left here, you actually have a fine needle aspirate from a lymph node metastasis in a patient. We can isolate these cells and grow them as if they were a cell line, reliably producing a wide number of cells over several weeks. Moreover, this enables technologies and experiments such on the right, where we're actually exposing a patient's own tumor to its own immune cells that's activated by immunotherapy, right? Allowing us to say that this patient will likely be an excellent candidate for immunotherapy before even treating them. We can only do this because we can grow their primary cells robustly without them senescing and actually acting as they would. Moreover, you can take this to another example where we're then sampling patients serially for certain rare cell populations, in this case, disseminated or circulating tumor cells. We actually see in the bottom left, we have a growing colony of the patient's own tumor cells that was extracted from 10 mils of blood, similar to them getting their cholesterol checked. We can then watch as this patient responds to therapy over time, and the fact that we can robustly grow these cells means that we have enough to do different genetic analyses, RNA levels, protein, and actually say, how is their tumor changing as they are being treated? And I think fortunately for us as a technology company, one of the big impetus is it's not enough for us just to show as an application that we can do it, but to be able to transfer that knowledge through successful and robust kits and consumables and products that allow others to accomplish the same things that we can do in our R&D setting. Fortunately, we've had quite a bit of success with that as well, where we have several different sort of early adopting customers, such as NCI here at UCSF, as well as with Bayer, in which they have been able to accomplish the same things we have in R&D, whether it be expanding primary patient cells, showing drug responsiveness being changed based on the environment which the cells are exposed to, or cultivating for these rare populations that people haven't been able to before. And so it's been a very exciting time for us, and I certainly appreciate the opportunity to share a bit of the work we're doing, and I'd be happy to take sort of questions later on in the session. Thank you.